Greetings, everyone. Thank you for taking the time to listen to this presentation. My name is Millie Dugal, and I'm a staff fellow in the Office of Medical Policy, CDR at FDA. I've been a member of the Cure ID team at FDA for a little over 4 years now, and my background and training is in maternal and child health. Drug repurposing is defined as finding new therapeutic uses for existing drugs. It is an innovative field that offers the benefit of having an existing medicine whose safety and dosing profile is well established. It's also faster and cheaper compared to traditional drug development. Drug repurposing seems to be a good approach for special populations. However, its use in this population has been limited. The Cure Drug Repurposing Collaboratory began its expansion into special populations, focusing on specific infectious diseases in pregnant women. There was a special focus into those diseases that have the potential to transmit the infection to the fetus, such as cytomegalovirus, toxoplasmosis, listeriosis, and so on. CMV was particularly interesting as it impacts both the mother and the baby, and there are no drugs approved for use in the perinatal context. We began having conversations with experts in the field, including researchers, academicians, and clinicians to determine the gaps in the knowledge of congenital CMV. The rest of my presentation will focus on the profile of the disease, testing and diagnosis, challenges in treatment, and the need to develop survey tools to address some of the challenges. Talking about congenital CMV, CMV is the most common congenital viral infection with both prevalence of 0.48 to 1.3%. In fact, according to CDC, about 1 out of every 200 infants is born with CMV infection. Transmission of the infection occurs when virus from the mother crosses the placenta and infects the fetus. During pregnancy, women may either have a primary CMV infection or non-primary infection, in which a previously infected woman experiences reactivation of a latent virus or reinfection with a new viral strain. Vertical transmission occurs with both primary and non-primary infections. Approximately 10 to 15% of infected infants experience symptomatic infection. And around 90% of babies born with congenital CMV present as asymptomatic, showing no obvious and visible symptoms of the virus. However, both symptomatic and asymptomatic newborns are at a risk of developing long-term neurodevelopmental morbidity, especially sensorineural hearing loss. There are no maternal vaccines or antivirals that can prevent congenital infection, so this remains an unmet area of need. As far as testing for congenital CMV is concerned, currently in the United States, there is no standardized approach to diagnosing congenital CMV infection. The diagnostic workup for newborns with clinical signs and symptoms suggestive of congenital CMV typically include CMV PCR from saliva, urine, or blood samples. This ideally must be done within 21 days of life to confirm congenital infections rather than those acquired postnatally. The absence of universal screening often leaves asymptomatic newborns undiagnosed. Pregnant women are not routinely screened either. Since symptomatic congenital CMV most likely occurs when a woman acquires primary infection during the first trimester, testing is not universal. Typically, serological assays are used, which have issues with false positives. Congenital CMV remains a challenging disease as there are no approved agents to prevent congenital infection. Moreover, there are no approved drugs to treat infants with congenital CMV, thereby remaining a great unmet medical need for these populations. There are treatment guidelines endorsed by American Academy of Pediatrics and recommended by the International Congenital CMV Recommendations Group. These include 6 months of valganciclovir for newborns with moderate to severe symptomatic congenital CMV disease who are able to start treatment within the first month of life. So we began having conversations with experts in the field of congenital CMV to explore how best CDRC could help bridge the gaps in this space. Our interest was to understand the knowledge gaps in the management of this disease to be able to determine a way to fill those gaps. We learned that there is a wide variability in the management of the disease, and at the outset, we don't know if valganciclovir is the standard of care at present. Valganciclovir also comes with its own set of issues, one of the primary ones being the length of its regimen. The drug has a long regimen of 6 months, which leads to questions about adherence to a lengthy regimen, toxicity associated with the drug, especially neutropenia, and the long-term follow-up required to understand whether the drug helped in managing the disease. There is also the issue of a gap between symptomatic newborns and those that are born asymptomatic, which leads to the issues of not knowing which newborn is being treated and for how long. Finally, we found that long-term outcomes in infants are not systematically captured and may only become clear after months or years when hearing loss or developmental impairment are noticed. So everything that I've presented so far leads us to question the need to develop survey tools for drug repurposing for special populations. We know that drug repurposing is an innovative field that requires systematic data collection using a patient-centered approach. We are aware that there are gaps in knowledge about the management and treatment of congenital CMV in neonates. At the outset, we found there may be variability in how different clinical practices treat the disease. So use of data from clinical experience can be of immense value in informing clinical trial design for repurposed or novel drugs to treat congenital CMV. The starting point for congenital CMV is to determine the various treatment regimens used across different neonatal practices. We believe that clinicians who treat these neonates and parents or caregivers who look after these children can be immensely useful in providing information about treatment, adverse effects, outcomes, and so on. A survey for clinicians or parents and caregivers can provide us with the information and a standardized tool that can help us analyze the responses to see the trends and various patterns of treatments and outcomes. So our team went ahead and developed a clinician survey for congenital CMV to be disseminated across various neonatal centers and among neonatologists. The survey is divided into 5 parts, and questions focus on the diagnosis of the disease, its management, antiviral treatment and safety monitoring, outcomes, and exploring opportunities for collaboration. CDRC's interest lies in partnering with stakeholders to address key knowledge gaps in the diagnosis and management of congenital CMV. If you're interested in providing feedback about the clinician survey or interested in helping us disseminate the survey to various neonatal practices, please email at millie.dougal@fda.hhs.gov. So in my last slide, I'm going to talk about the various ways in which CDRC can address this issue. Since there is no standard screening in pregnant women, CDRC will focus presently on neonatal rather than maternal infection. We will reach out to neonatologists to share their experience treating infected infants using the Cure ID app, collecting details about the drugs used and regimens, and collect additional information through the clinician survey. We can leverage Cure ID to collect real-world data for treating congenital CMV in order to generate testable hypotheses to inform clinical trial design. We are also considering using the patient portal for parents to share the long-term outcomes for neonates who are treated. Thank you for listening to my presentation and for your support to this program. I look forward to discussing some issues on the panel. Thank you.